Hello everybody and welcome back to Creation Myths. I have a really fun one for you this time. We are back talking about creationist contradictions, and this time we are pitting two of the big names in young Earth creation science against each other. Today we are talking about Dr. John Sanford versus Dr. Nathaniel Jeanson. The arguments we are going to be talking about today are genetic entropy proposed by Dr. John Sanford and the created heterozygosity and natural processes model of Dr. Nathaniel Jeanson. Now, I have talked about genetic entropy and created heterozygosity at length before. Both of those videos are linked down below. So if you want to see why each of these creationist arguments is wrong, check out those videos. Remember, the purpose of this series is not to debunk each of the arguments we're talking about. Instead, it's to show how the creationist arguments are incompatible with each other. So today, we are going to show how genetic entropy and created heterozygosity are incompatible with each other, despite many professional creationists accepting both of them simultaneously. In order to show why these two arguments are incompatible with each other, we need to introduce some basic genetics terminology. The first term we'll do is diploid. The human genome is diploid. That just means we have two complete sets of chromosome, one set from mom and one set from dad. Because we have two of each chromosome, that means we have two of each locus in our genome. Locus just means a specific location somewhere in our DNA. And within the human genome, you have two of each locus because you have one on each homologous chromosome. Homologous chromosomes are pairs of chromosomes that correspond to each other that you got from each parent. So chromosome number 1 from mom and chromosome number 1 from dad are homologous chromosomes. And on that pair of chromosome 1, we can find each locus twice, once on mom's copy and once on dad's copy. Now, when we compare loci to each other, loci being the plural of locus, we can describe them in a couple of different ways depending on whether they're the same or different. When two loci on homologous chromosomes are the same, the word for that is homozygous. That just means the sequence at that specific location on the chromosome is the same from mom as it is from dad. So in this example, we're just using capital letters to show that for region A or locus A or gene A or whatever this is, you get one sequence from mom and you get the same sequence from dad. So for this locus, these homologous chromosomes are homozygous. The opposite of homozygous is heterozygous. That's when two loci on homologous chromosomes are different from each other. So for gene B or region B or whatever this is, you got the capital letter version from mom and the lowercase letter version from dad. That means for this region, these homologous chromosomes are heterozygous. They have different sequences at that site. One more important thing to keep in mind today as we go through these two competing hypotheses is that as you increase variation, that is, you have mutations that occur and change the sequence at specific places on your chromosomes, that results in an increase in heterozygosity, because when you have a change on one chromosome within a homologous pair, if you don't have that exact same change at the same site on the other chromosome, that's going to result in a heterozygous state for that particular site on the chromosomes. So I think that's all we need for background information. Now we can get into these specific hypotheses, and we'll start with Dr. John Sanford and genetic entropy. Dr. John Sanford is a plant geneticist, and he coined the term genetic entropy in his 2005 book, Genetic Entropy and the Mystery of the Genome. The genetic entropy hypothesis works like this. He claims, he's wrong about this, by the way. This is wrong, see the link below. But Sanford claims that virtually all mutations are inherently harmful independent of the context, and that such harmful mutations inevitably accumulate within genomes over generations. This results in a fitness decline and ultimately in extinction in whatever population it is you're talking about. Sanford describes it like this. He says, when selection is unable to counter the loss of information due to mutation, a situation arises called error catastrophe. If not rapidly corrected, this situation leads to eventual death of the species, extinction. In other words, if we were to simplify this a bit, the idea with genetic entropy is that you start with a mutation-free created genome. Mutations occur and accumulate because there's no way to get rid of them. This causes fitness to decline, and eventually extinction must result. In fact, Sanford chose the term entropy very deliberately due to the inevitable nature of this process, he claims. He wrote this: "Mutational entropy appears to be so strong within large genomes that selection cannot reverse it. This makes extinction of such genomes inevitable. I have termed this fundamental problem genetic entropy." So this is a fundamental, inescapable process based on mutation accumulation. Keep that in mind as we look at Jeanson's hypothesis. Dr. Nathaniel Jeanson has proposed the CHNP model, or created heterozygosity and natural processes. Dr. Nathaniel Jeanson, of course, is the Harvard-educated developmental biologist who works currently, as I record this, at Answers in Genesis. He's written about this model in a number of places. For example, he published in Answers Research Journal, the fake AIG in-house peer-reviewed journal, in this piece called On the Origin of Eukaryotic Species, Genotypic and Phenotypic Diversity, Genetic Clocks, Population Growth Curves, and Comparative Nuclear Genome Analysis suggests created heterozygosity in combination with natural processes as a major mechanism. That is a mouthful of a title, but basically in this piece, co-authored with Dr. Jason Lisle, he explained this CHNP hypothesis. He also wrote about it in some length in his book, Replacing Darwin. Here's right from the beginning of chapter 10. He wrote, "Most of their traits seem to have been present in coded, genetically heterozygous forms in their ancestors. In the descendants of these ancestors, shifts from heterozygosity to homozygosity would have been easy. These shifts would have revealed traits that were previously hidden. For example, the genes controlling zebra stripes likely come in dominant and recessive forms. As offspring arose with homozygous forms of one or the other, distinct traits would have appeared, striped and unstriped individuals. Population subdivision via migration would have isolated these distinct traits and promoted the rise of new species such as zebras and asses." That's a bit of a mouthful, so here is a simpler version. Under this model, new species tend to arise via shifts from more heterozygous nuclear DNA states to more homozygous nuclear DNA states. In other words, we can simplify this. We could say kinds, the created kinds, were created in highly heterozygous states. They lose heterozygosity as they radiate around the world geographically, and this results in speciation from those created ancestors. The more homozygous descendant groups are the extant species. Those are the things that are alive right now, and again, it's due to the loss of heterozygosity since creation. Now, an important caveat here is that virtually all existing variation was present in this initial highly heterozygous state. Jeanson writes, "But if evolutionists can invoke pre-existing differences, why can't creationists? If evolutionists explain 88% of our differences as pre-existing, is it too much of a stretch to bump the number up to 99.6%? So the idea here is that the vast majority of the variation we see in extant species is due not to processes like mutations, but it's due to recombination from that initial heterozygous state. It's important to note here that prominent creation scientists accept both of these models. Here, for example, is Dr. John Sanford and a frequent co-author of his, Dr. Rob Carter, who is affiliated with CMI, Creation Ministries International. Here's a piece, I think this was a report from a conference called Adam and Eve, Design Diversity and Allele Frequencies. And in this piece, they basically argue in favor of created heterozygosity, right? They say, yet Adam and Eve could have been created massively heterozygous, and go on to say how, they are proponents and defenders of this idea. Here's a piece they co-wrote together. This is actually a real paper published in Theoretical Biology and Medical Modeling. I've talked about how terrible this paper is, but it's an attempt basically to show that genetic entropy happened in H1N1 influenza. So here we have Carter arguing in favor of created heterozygosity, and then also co-authoring a paper with Dr. John Sanford on the topic of genetic entropy. Sanford, again, wrote the book on genetic entropy, also proposes and defends created heterozygosity. So professional creation scientists hold both of these hypotheses as valid. So now it's time for part two. Fight fight. Let's see why these two hypotheses are completely incompatible with each other. Let's just do a quick direct comparison, and then we'll go into a little more detail. So we have Jeanson and CHNP. Starts with a highly heterozygous state and then loses heterozygosity over time. Sanford and genetic entropy, because it's based on mutation accumulation, says that you're going to gain heterozygosity over time. Jeanson's model has almost no role for mutations. Remember, according to his model, over 99% of the variation is based on those initial created states, where Sanford says that genetic entropy, it's all about mutation accumulation. It's completely driven by mutations. And then finally, Jeanson sees variation as potential for speciation. Because you can have lots of viable states in your initial heterozygous created state that are then going to separate from each other, you have many variants that are perfectly fine. And if you have mutations that cause you to switch from one of those to another one, those mutations are perfectly fine. They're not harmful at all. Sanford sees mutations very differently. According to Sanford, any variation, any mutations are deviation from that initial mutation-free created state. And according to Sanford, as I'll show you in a few minutes, virtually all the possible variation from that state is actually harmful. So you can see right here just how starkly these models are contradictory with each other. But we're going to go into more detail for each of these points, starting with heterozygosity. So Jeanson's model, as we've been saying, starts with a ton of, they say, front-loaded diversity, created heterozygosity, and then you lose it over time. So here we have just a simple representation of the created kinds that have a bunch of variation built in at creation. But as you have reproduction and you have geographic isolation, descendants from those created kinds are going to lose that created variation. And the extant species that we see today, instead of having lots of heterozygosity, are, according to Jeanson, to be found in highly homozygous states. And it's this transition from heterozygosity to homozygosity that's actually driving the speciation. Now, again, I'll say it again, just to make sure we're all clear, mutations play almost no role in this process. 99.6% of the variation, according to Jeanson, is present in that initial created kind. Compare this with John Sanford. According to Sanford, you start with a specific mutation-free state. And mutations are going to occur and accumulate over many generations. This is going to increase variation, and therefore it must necessarily increase heterozygosity. And we could illustrate this just by looking at a pair of homologous chromosomes. So here we have, a chromosome from mom and a chromosome from dad. There's going to be some differences between them, but there's going to be a lot of places where they're the same. Well, if a mutation occurs at one of those sites where they're homozygous, they're now heterozygous at that site. So this orange change indicates a mutation where this chromosome now has that variant, this chromosome doesn't. That's new variation that is now increased heterozygosity. Over generations, you're going to have more mutations accumulating in these chromosomes, leading to an increase in heterozygosity between these homologous chromosomes. So according to Sanford's model, you're going to see an increase in heterozygosity from creation to the present. Now remember, mutations are undirected. Mutations are probabilistic. So these two chromosomes, the homologous chromosomes, the one from mom and the one from dad, they're going to mutate independently of each other. So I want to make it very clear, as mutations occur, that means heterozygosity is necessarily going to increase. There's no way for Sanford's model to work that doesn't result in an increase in heterozygosity. So that is a direct conflict with Jeanson's model. Now let's look at how the two models treat mutations. As we've seen, Jeanson has virtually no role for mutations in his model. He writes mutations might increase the diversity slightly, but around 99% of the DNA differences between chromosomes would be due to inheritance, which is to say the initial created variation. And these differences should be easily classifiable into one of the four starting chromosomal versions in the original parents, which is to say the male and the female of the original created kind, each of whom are diploid, so you have a total of 4 chromosomes there. He's saying over 99% of the variation can be traced to those initial 4 chromosomes. Sanford, on the other hand, says mutation accumulation is inevitable and catastrophic. He writes, describing kind of the consensus in the field of genetics, that there are at least 100 nucleotide substitutions, he calls them misspellings, whatever, per person per generation. That's in the human genome, 100 mutations per person per generation. And that's basically correct. He goes on to write, given these numbers, there is no realistic method to halt genomic degeneration, which is to say, halt mutation accumulation. So in Sanford's model, mutation accumulation is the driving mechanism, and the accumulation of mutations is playing a huge role in the extant variation that we find. Finally, let's look at how these two models treat variation and fitness. According to Sanford, virtually all mutations are bad. Recall that he proposes kind of a mutation-free created state, and he writes, it can very reasonably be argued that random mutations are never good. In other words, any deviations that occur from that created state since creation are going to be harmful or degeneration or hurt fitness in some way. In contrast, Jeanson proposes a highly heterozygous created state followed by loss of heterozygosity as lineages diverge from each other. This implies that there must be many approximately equally fit variants that are possible from that highly heterozygous created state. Jeanson actually writes that heterozygosity, the created variation, represents speciation potential. He says that high heterozygosity, i.e. low homozygosity, represents high potential for subsequent speciation. Low heterozygosity, i.e. high homozygosity, represents low potential. The process of speciation that we've derived naturally dilutes the speciation potential in descendants. In other words, what Jeanson is arguing here is that within this created heterozygous state, you have a lot of variants that would work just fine in this lineage or that lineage. And as they geographically separate from each other, they're going to lose variation, they're going to become more homozygous, but they're going to do it in different ways. They're going to basically have different subsets of that initial created heterozygosity. That means that the variants that are front-loaded in Jeanson's model must be more or less okay, all those different variants that represent over 99% of the existing genetic diversity. But if that's the case, that means that if one lineage has a mutation that turns it into the variant that's present in another lineage, then that can't be a harmful mutation. And because Jeanson says over 99% of the extant variation was present in the created state, that means that the vast majority of mutations are actually perfectly fine, according to Jeanson's model. That is a direct contradiction between the two, and it's just another reason why these two models are completely incompatible with each other. So to summarize this creationist contradiction, we have been looking at genetic entropy from Dr. John Sanford and the CHNP model from Dr. Nathaniel Jeanson. These are two of the big names in young Earth creation science and two of the big models in young Earth creation science. They're so prominent and so important to young Earth creationists that many young Earth creationists accept both created heterozygosity and genetic entropy as valid models. But as we've seen, these two models are directly contradictory in several ways. They are contradictory in terms of how heterozygosity changes over time, in terms of the role of mutations since creation, and the fitness effects of potential variants. The big takeaway here is that it is impossible to reconcile these two models. Young Earth creationists have to pick either created heterozygosity or genetic entropy. You can pick one, you can pick the other, but you can't pick both. Thank you for watching. If you enjoyed this, found this informative, please hit the like button, subscribe to the channel if you haven't already subscribed, and leave a comment. Thank you for watching. Don't get fooled.